Bonjour tout le monde. Après un petit faux départ, on commence vraiment. Désolé, on aurait pu faire des pas de danse pour
Hello,
continuer.
everybody.
Donc,
There
je suis
was
ravie de
a
vous voir
false
tous
start.
aujourd'hui.
I'm
Merci
very
pour votre
happy to
présence.
see you all today. Thank you for being present. This is the episode number 2
Il y a 4
in the
ans.
fourth
Le France
edition of Parlons
Parkinson
Recherche. 4
organise
years now that France
ce projet.
Parkinson
Le
has been
projet
organizing this event to
Donc, la semaine
share with
passée,
you the
on était à
projects we
Nantes
support.
et on a
Last
parlé
week
de
we were
recherche
in
clinique.
Nantes and we talked about
Et pendant
clinical
l'événement de Nantes, on
research.
nous a demandé
And during
si on
the
faisait des
Nantes
collaborations
event, they
avec
asked us whether we were doing something about the
C'est ce qu'on va
causes
faire
of the
aujourd'hui.
disease and preclinical
Aujourd'hui, je
research.
suis
And this
ravie
is
de
what
vous
we're going to
rassurer
do
sur
today. The
Parkinson dans les
subject today will be Parkinson
laboratoires
in the
aujourd'hui, on a
laboratories. We're
4
very lucky
chercheurs
that
et
we have
chercheuses
4 researchers
impliqués
with us that I'm going to introduce a little later. I just wanted to tell you that today is going to be a slightly different episode because we're doing this in collaboration with the Foundation for Research on the Brain. We've been members of this foundation for years, and it also funds research that France Parkinson is supporting. We also support projects that have been chosen by the scientific committee. And today we're going to hear about 2 projects supported by France Parkinson, which were chosen by the scientific council of the foundation, sorry, says the speaker for clinical research. Now, in this episode, we're also supported by DIMSIM Brain. What is DIMSIM Brain? DIM are major innovation research domains, which are supported by the Île-de-France region, where we find numerous laboratories. And in the funding, they support events for scientific dissemination of science. And I would like to thank DIMSIM Brain for supporting us today. As I was telling you earlier, we have 4 researchers today with us. I will introduce them very quickly in the order in which they will be speaking. Christelle Bonez, who is at the CNRS in Marseille. She's a research director for CNRS in Marseille. Dr. Hélène Cheval, who is a lecturer at the University La Sorbonne and researcher in the Institut du Cerveau in Paris, Brain Institute. Professor Pascal Fossat from the Bordeaux University, and is a team leader at the Institute for Neurodegenerative Diseases in Bordeaux. And finally, Louise-Laure Mariani, who is working for the La Pitié-Salpêtrière Hospital and researcher in the Brain Institute as well. Before they take the floor, just a few words regarding the way we're going to organize the meeting. We will listen to the 4 presentations. You see that there are QR codes everywhere. So if there's anything you don't understand, between every presentation, we will be asking you questions, you in the audience, to check whether you have been listening correctly. And it's just a few questions between the speakers, and you will see the answers coming up on the display on the screen. Once the 4 speakers have made the presentation, there will be 1 hour for exchanges with the room. And both people following us online and people here in the audience will be able to ask questions. There are no good and bad questions. It's not a private medical appointment. Don't ask personal medical questions, but ask questions about things you may have not have understood during the presentations. And we will share the time between the people online and the people in the audience. Without further ado, we will listen to the first speaker, Dr. Christelle Bonès, who's going to tell us about subthalamic nuclei. Am I supposed to activate? Oh, sorry. My apologies. I'm going to talk about a story on the role of the subthalamic nucleus and the action in social disorders. I would like to start by reminding you of the context that we find ourselves in when we talk about subthalamic nuclei. It is the deep brain structure that we target with deep brain stimulation treatments when we're dealing with Parkinson patients. This was evidenced and published for the first time by the group of Professor Benabi in Grenoble. And they showed motor improvement in the patients. It was actually spectacular. When it works really well. We're talking 50, 60% motor improvement in some patients. For us, the challenge is when we talk about research in the animal, is to make the technique accessible. And in this case, we spent time introducing big electrodes that you see on the left-hand side, and we moved to smaller electrodes to adapt them to the size of a mouse or a rat. The section image that you see here is about the size of a 20 cents of euro coin. It's a very, very small area in the rat's brain that we are targeting. So we have to have tiny electrodes and we have to adapt the stimulation. Like with our patients, we have to adjust the parameters for every single individual. Now, regarding the mental process in which the subthalamic nucleus is involved, we know it is involved in motricity, but when we looked at the books, the manual, the handbooks, the conventional circuit of reward, subthalamic nuclei are not involved. So they're not there, they're not to be found. So I added them based on the literature data explaining how it's connected. It receives direct information from the prefrontal cortex and from the dopaminergic neuron projections, and it then is forwarded to the subthalamic nucleus, which means that the subthalamic nucleus is there acting like the rewarding center, the nucleus that acts as the rewarding center. And we thought, well, there's no reason why we shouldn't think that the subthalamic nucleus is involved in the process. This was evidenced. Data was collected and published in the literature confirming this assumption. And then we started looking into motivation dysfunctioning. And the most conventional disease when we talk about reward dysfunctioning, it's addiction, because here in this circuit there are several stages. Motivation increases for the abused substance, then there is loss of control regarding the consumption, and progressively the most affected subjects will do what we call compulsive consumption. In fact, in spite of the fact they are aware of the negative consequences, they will still do it. For instance, for those of you who smoke, on the cigarette pack, it's written smoking kills, but you still consume the cigarettes. But that's exactly what I mean by compulsive consumption, followed by abstinence periods, and then there will be a relapse most of the time. And these aspects, when you manipulate the subthalamic nucleus in the rat, we have been showing that there are beneficial effects in as far as we can stop the addiction process in the rats in the several stages by manipulating the subthalamic nucleus. On the other hand, it also is involved in emotional processes, which was shown in Parkinson patients. For instance, here, the absence of emotional feeling, perception, the difficulty of perceiving and recognizing emotions on photographs. This has been confirmed in rats. The combination of both. Why am I telling you this? Because we are interested by the way in which drug abuse can be regulated by the presence of other individuals. We are trying to understand what substrate in the brain can carry the social interaction. And we thought that the subthalamic nucleus is very useful because it plays a role in addiction and in emotional processes, so that the presence of a cage mate or another individual can generate emotions. So we started looking into this, and what did we find? Here we have manipulations. There's a rat, there's a grid, and there's another rat on the other side of the cage watching the rat take a drug. And when they're on their own, you see the consumption on the left. And on the right-hand side, the graph shows what happens when there is another rat next to the initial rat. If it's a rat, a cage mate they're used to, nothing happens, consumption doesn't decrease. However, if the rat is not a cage mate, is a stranger rat, then there is a decrease in the consumption, possibly because the fact that there is an unknown rat next door to them, they are not stressed, and they don't increase the consumption, but rather they decrease the consumption. When the cage mate is present, there is no decrease if the cage mate is well known. And that is interesting because there are two rats in a cage, and when they are together, always one is dominating and the other one is being dominated. So we try to understand the influence of dominance in the relationship. The next graph is very complex, but follow me, I'll guide you. You look at the green dots. These are animals which are dominated. And behind the grid, they have the other rat who is the dominant one. Obviously, an unpleasant situation, but the consumption here does not decrease. The yellow dots correspond to the dominating rats who see the dominating rats seeing the dominated rat on the other side of the grid. And here the cocaine consumption will decrease. If we remove the subthalamic nucleus, we see that there is an even more drastic decrease in cocaine consumption. And that consumption is not dependent on the dominance relationship. It's as if there was no more difference between the dominated and the dominating rat. So it raises questions regarding the involvement of subthalamic nucleus in the recognition, the acknowledgement of the other individual behind the grid. And then we looked at the network because the subthalamic nucleus cannot do this on its own. In the brain, all cerebral areas are connected. So we started to question, to ask what way could modulate the social interaction in drug consumption. We made a number of projections from the anterior insular cortex or the prelimbic towards the subthalamic nucleus. And then we have some very specific tools that allow us to target selectively some areas and inactivate only those projections that go from the anterior insula to the subthalamic nucleus or only from the prelimbic to the subthalamic nucleus. In blue, we see the performances of the control animals. Here, nothing is inhibited, and we put them in the basic situation. 1B, 1A, M is on they're in presence of a male. A1, they're on their own. A2, they're alone again, and F, they're in presence of a female rat. And you see that the control animals, when they are in the presence of a male, there will be a decrease in the cocaine consumption, and there will be a rebound effect once they find themselves alone again. However, the presence of a female cage mate does not have the same impact. Same conditions, if we inhibit the projection of the anterior insula towards the subthalamic nucleus, here we see there is a slight decrease in the cocaine consumption if we inhibit the projection from the anterior insula towards the subthalamic nucleus. However, this is exacerbated by the presence of a male rat and not a female rat. Sorry, says the speaker, I got it the other way around. There's decrease also in presence of a female rat. So the anterior insula will need to decrease in the presence of both a male and female rat. If we inhibit the projection from the prelimbic towards the subthalamic nucleus, we see that the consumption decrease is inhibited during the pathway inhibition, condition A1. However, this is not modulated by the presence of a male rat or a female rat, which makes us believe that maybe it is the anterior insular subthalamic nucleus pathway that carries the effects we see when we inhibit the subthalamic nucleus directly. Next questions that we asked ourselves in this contract: what is the role of the subthalamic nucleus in social motivation? Does it have anything to do with motivation and interest for the other individual behind the grid? What impact does this have on the modulation? So we developed a task in which the rat is no longer consuming cocaine and must push a lever to open the door. You see the door here going up and down, and the rat has access through the grid to the other individual. He can interact with the other individual behind the door. By doing that, we can modulate the various conditions. We can test female rats and male rats and see how they push the lever to have access to another individual who is either a cage mate or a stranger rat. In control animals first, that we see how the gender can influence the results. If the female is pushing the lever, every time it pushes the lever, the door opens and we see the green dots. Female, and the blue dots are the male. Green dots are the female. And we see that the male rats are not working as much as the female rat to have access to the other rat in the cage behind the grid. If we look at the influence of familiarity, here we have male rats, so it's light blue color. Male rats working to have access to an interaction with the other rats behind the grid. We see a decrease with time, as if they sort of became bored of needing to push a lever to have access to an individual which they already know. Whereas in dark blue, we see that animals who work to have access to a stranger rat, although the activity here is greater and does not decrease. What is the influence of dominance? Because again, we're looking at rats who have to accomplish a task to have access to the other rat in the cage. And we see the difference here depending on the dominance relationship. Dark purple curves show that the dominant animals will continue working with the same ardor to have access to the other rat in the cage, who is the dominated rat. However, dominated rats, light purple, will become bored and will stop or decrease the number of times they push the lever to open the door and have access to the other rat, who happens to be the dominating rat, the dominant rat. It makes sense. But what happens if we inhibit the subthalamic nucleus on the social interaction interest? In blue, we have the male rats pushing the lever to have access to the other rat in the cage. And over time, this decreases, but it is compensated for by the inhibition of the neurons in the subthalamic nucleus in orange. So we see a stabilization in time. What happens in the dominated rats who limit the number of times they push the lever? Purple curve. We see that the decrease is also compensated for by the inhibition in the neurons of the subthalamic nucleus. As if the inhibiting the subthalamic nucleus allowed to keep a certain interest for the other rat in the cage, regardless of who that rat is. Now we have increased the effort to be made on the lever to open the door. Until now, they only had to push the lever once and the door would open. So to understand whether they are really motivated, we're asking them progressively to have to push twice, and then 3 times, and then 4 times, and then 10 times for the door to open. And we look at how many times they will push the lever to have access to the other animal behind the grid. Female rats seem to have a relatively stable motivation that will last in time, 2 blocks of 5 sessions during 10 days, consecutive days. In the male rats, again, we see a difference between stranger rats, which will keep a relatively sustained level of motivation, but we see a decrease in the motivation to push the lever to have access to the other rat in the cage. And if we inhibit the subthalamic nucleus in this task, we see a decrease, including in the motivation to work to have access to another rat when we inhibit the subthalamic nucleus. And if we're looking at cage-mate rats, we see that the decrease observed in control rats, light blue. This is the decrease over time which you observed in the previous graphs, but in orange, we see that when we inhibit the subthalamic nucleus, we actually exacerbate the decrease. In conclusion, I have just told you that the subthalamic nucleus plays an essential role in addiction, and it's by inactivating it can be beneficial. Now, that's something we already had observed in the Parkinson patients who suffer from dopamine deregulation, dopaminergic deregulation. They take L-DOPA in an abusive way, and deep brain stimulation may be beneficial on those patients. The subthalamic nucleus also plays a role in cocaine intake modulation when another rat is present, and this is possibly due to the fact that it involves the pathway coming from the anterior insula towards the subthalamic nucleus. Finally, the subthalamic nucleus plays a role in social motivation. Its inhibition will decrease social motivation, and it decreases the boredom effect induced by the partner, which should make the people accompanying a patient today happy. And obviously, this presentation is open to discussion and open for discussion. And I would like to thank the Trans-Parkinson Federation for their support and all the members in my team. Thank you very much.
Merci beaucoup.
Thank you.
Je pense que le noyau sous-thalamique n'est
I
plus
think that
de ce fait.
subthalamic
On va passer à
nucleus
des
no longer
questions.
holds any
En fait, par
secrets.
rapport à
We'll
ce
move
qui
to
est
the
raconté, qui
questions
vous sont
on
destinés. Donc,
this
on va répondre au QR code.
presentation.
Si on peut avoir la première question, s'il vous plaît.
Can we have the first question?
Les questions, on répondra aux questions
No, we will
des
answer
orateurs
your
dans la deuxième
questions
partie
at
de
the
la
end,
conférence.
but right now we're doing
Donc,
questions
en
to
plus du
you, to
mouvement,
the audience.
parce que c'est bien connu dans le
So,
mouvement, le
on
noyau
top of the
sous-thalamique
movement,
joue un
the
rôle dans, je
subthalamic
vais choisir,
nucleus
la
plays a role
motivation
in
sociale et certains
social
comportements liés
motivation
à
and
l'addiction,
some
le sommeil profond
addiction-related
ou la
behaviors,
régulation
deep
hormonale.
sleep, or hormonal regulation. Okay?
Ah bah, je confirme que vous
I
avez bien compris.
confirm. You
Donc, c'est
all seem
intéressant
to
de
have
voir
understood. It's
qu'on
very
étudie
interesting.
au-delà toujours de simplement les symptômes moteurs. Il y a beaucoup de choses qui
We
sont
are
aussi
not
importantes
simply looking at
dans
motor
ces
symptoms.
expériences
There are other
individuelles,
things
mais
that
qui
we are
peuvent
studying, and that's
permettre
precisely
d'avancer en
why these
connaissance
projects
aussi.
are very helpful to improve knowledge.
On a une deuxième question
There is a second question,
qui sera plus un nuage
but it's
de
more
mots. Donc, en un
a
mot,
word
avant
cloud.
de décrire un
Give
mot,
us
si le noyau
one word.
sous-thalamique était un personnage,
If the
ce serait
subthalamic nucleus
quoi?
were
Ce serait
a
qui?
person, a character,
Est-ce que
who
vous
would
avez
it
un mot
be?
qui vous
Do
vient
you
à
have a
l'esprit par
word
rapport
that
à ce que
comes
vous
to
avez
your mind?
entendu
Just give
aujourd'hui?
one word.
Et si vous pensez au
If the
noyau
subthalamic
sous-thalamique?
nucleus were a character, who would it be?
Ah, c'est plus difficile, hein?
It's a slightly trickier question.
Oula! Donc
Okay.
vraiment ce qui vous passe par l'esprit, hein.
Whatever
N'essayez pas
comes to
de,
your
il n'y a pas de
mind.
réponse
There are no good
bête
and
ou
bad
intelligente.
answers
Toutes les
or
réponses sont
stupid
bonnes, c'est
and
pour
smart
moi, c'est
answers.
pour comprendre un peu.
Just
Euh,
whatever comes to your mind.
professeur,
Professor,
tu pourras retenir les mots.
can you remember? Can you try to remember the words?
Professeur a quand même
It
une
seems
grande
that
place, mais on voit
professor or
beaucoup de choses. Il y a
teacher
manipulateur
takes
quand même.
precedence. Manipulator as well.
C'est assez intéressant de voir toujours les mots qui nous
Very
viennent qu'à
interesting
l'esprit, surtout si on ne doit pas
to see
réfléchir
what
et
comes to
qu'on
your
écrit le
mind
premier mot qui nous passe
just
par la tête.
spontaneously. If you just write the first word that comes to your
Après, c'est
mind
clair, quand il y a les mots qui sont
spontaneously.
là, ça nous donne des idées. Des fois, on répète
And,
le
you know,
même,
it gives us an
mais
idea.
c'est
Obviously, you
intéressant.
see the words on the screen, so maybe you're encouraged to
Là, vous
maybe
voyez
write
qu'on a quand
whatever
même
you see on the screen,
le
but
chercheur qui prend des
it
photos, hein.
seems that there are researchers taking pictures.
Donc, merci pour
Thank
voilà, de
you
prendre le temps
very
pour ça.
much for having
On
taken the
garde tout
time
ça en
to answer
mémoire.
the
Bien sûr,
questions.
on
We
pourra en
keep
discuter
everything in
après.
mind and we can
Merci
discuss
pour
this
votre
later.
soutien régulateur et professeur.
Regulator and
On va passer à notre
professor.
prochain
Okay, we
orateur.
move on to the next speaker.
Donc, il y a un jingle pour présenter tous les personnes en ligne.
Bonjour, je voudrais commencer par remercier France Parkinson pour m'inviter, pour présenter mes recherches et aussi pour
Hello, I'd like to
vraiment
thank
organiser
Franz
un événement
Parkinson for
sur ce
having
sujet
organized
qui est
this
très
research.
important et puis pour participer au
It's very
financement des
important
recherches euh, pour nous et pour nos étudiants.
for us and
Alors moi,
for
je vais
our
vous parler
students.
du projet qui est justement financé par
I would
France
like to
Parkinson,
discuss
qui
the
est
project
comment
being funded
comprendre
by Franz
comment
Parkinson. How do we
essayer de
understand
comprendre
how Parkin protects our cells from stress with the organoid model.
Understanding how Parkin protects our cells from stress in mitochondria using the organoid model. We're trying to understand how this degrades into predominantly in Parkinson's disease. Why do dopaminergic neurons degenerate? The appearance of motor dysfunctions of significant proportions. There are several hypotheses that have been put forward by many scientists in the world. One of those is that the mitochondria, which are energy-producing factories of cells, are not working properly. There's a defect in energy production. There are a certain number of indices which lead to that idea. For example, in human samples, we realized that there are parts of the energy producing factory, as it were, of mitochondria that are not working right. And there can be toxic agents, such as retinol or MPP+, which target parts of the energy production activity in mitochondria. And these agents can bring about symptoms that are reminiscent of Parkinson's disease. Lastly, there are forms, genetic forms of familial type of mitochondrial proteins involved, such as PINK1 or Parkin. These genes code proteins that are in mitochondria that are involved in mitochondrial quality control, including mitophagy, in other words, in familial form of Parkinson's. So they can actually feel when the mitochondria are not feeling well. And they will be indicating to the cell, okay, now it is time to degrade the damaged portion to get rid of it and to keep the healthy part. And we call that mitophagy. We believe that if dopaminergic neurons intervene, it's because they have an increased vulnerability to stress. There's a bit of a dysfunction due to the mitochondria. But there are two questions. The first is that the molecular players identified to date, PINK1 or Parkin, are present in all cells, not only in dopaminergic neurons. However, in Parkinson's, it is mainly the dopaminergic neurons that generate. But there are molecular mechanisms specific to these that cause these neurons to be particularly vulnerable to mitochondrial stress. So what are the mechanisms proper to these dopaminergic neurons, which make them more vulnerable to stress, but we don't know why it's different. We don't know those mechanisms. In the same way, PINK and Parkin are stress sensors. They're precocious. We can wonder if these are dysfunctioning, as in the case of mutation. Does this automatically bring about degeneration of DA neurons in Parkinson's disease early on in life? But they're silent because you don't realize that they're there right away. We are trying. Christiane Duc, who's a doctoral candidate in the laboratory. If you'd like to meet her, there's a poster of her here. We're trying to understand how Parkin plays a role in protecting cells faced with this type of dysfunction of mitochondrial stress, and if the role is different depending on the cell type, what means that the neurons are more or less sensitive to this. We believe that Parkin and PINK act as a mitochondrial stress sensors, and they can coordinate various protective responses in cells, not only mitochondria, but a whole set of responses to protect the cell from these stress. So Christian's objective is to identify the molecular mechanism triggered in dopaminergic neurons following mitochondrial stress, and to evaluate the dysfunction of these stress responses and see if they're different in other cell types that we have in the brain, and obviously to assess the dysfunction in the context of the Parkinson's disease, for example, in the absence of Parkin. So to do this, we use a study model. It's quite recent, which we call mini brains. They're more organoids that we obtain thanks to stem cells that are pluripotent, and they are induced pluripotent stem cells during development. In the uterus, we have stem cells that are embryonic, that have the power to become any type of cell. They can become, as you can see, over time, they will be differentiating into different types, and little by little, these cells will be specialized to become skin, pancreas, lung, and even in the brain, there are many different types of cells. So this is differentiation. Now, and it's only recently that we are able to go backwards. In other words, starting from these specialized cells, we can reprogram to back up and become some cells that are pluripotent. In other words, they have the power to re-become many other things. And yes, it's induced because we program them that way. So in practice, how does it work? Well, we take a skin or a blood sample from the patient. And based on that, we reprogram to have stem cells that are pluripotent from the patient. And these stem cells, we can then if in the presence of a certain milieu, you can obtain dopaminergic neurons or skin cells or heart cells or whatever. We try to obtain cerebral tissue, and we try to obtain something that is close to the region that gives notion to black matter, the ventral mesocephalus. What you see on the left, like the size of lentils, these are the organoids. And we are able to cultivate these cells so that they get self-organized into 3 dimensions, and they're adopting an organization similar to that which we observe in different regions of the brain. There's an example in the middle. This is a cross-section of an organoid. You see a superficial layer, and you see at the beginning of the development in the cortex. And the advantage of this method is that we will obtain neural networks that are much more mature than when you do 2-dimensional cultures. This approach, using these organoids, makes it possible to do many things. This is a modulatable technology because using these induced pluripotent stem cells, we can generate organoids from different regions of interest. We can have organoids from the central ventral midbrain organoids, where the substantia nigra originates, or subcortical organoids from the striatum, etc. And with blood samples or EPS, we can obtain brain immune cells. And with all of these, we can assemble these different regions, and we call these assembloids. The idea being to see, for example, if these regions can be connected as they do connect in real life. In this context, Christian, who is studying the response of dopaminergic being exposed to stress, she uses
Et donc, elle a pris des cellules
took
pluripotentes induites,
pluripotent,
contrôle ou qui n'exprimaient
induced
pas la Parkin,
pluripotent cells,
et elle
which
les a
were not
cultivées
expressing
de
Parkin,
façon à
and
ce qu'elles
she
deviennent des
cultivated them
organoïdes, soit
so
en
that they
condition
become
basale,
organoids,
soit
either
en
in basal
condition
condition
où on leur
or
fait
in
subir un
chronic stress
chronique, c'est-à-dire un
condition.
petit stress
In
régulier,
other words, a
où on
bit of a
va
regular
faire
stress where we
dysfonctionner la
are going
chaîne
to
de
help
fabrication d'énergie.
the
Un
manufacturing
petit peu,
chain
mais de façon
to
constante. D'accord. Et
malfunction
au
a
bout de
little bit,
60
but
jours, c'est-à-dire 2
constantly.
mois
And
de
after
culture,
60 days, in other
eh
words,
bien,
2 months of
bah,
culture,
Christiane, elle a fait pas mal
she
d'expériences, des
has done a
marquages
lot of
fluorescents,
experiments about
mais
fluorescent
aussi,
labeling
et je vous expliquerai après, en fait, des techniques très poussées
and
pour regarder à
various
l'échelle
highly
de chaque
developed
cellule
techniques for each
quels
cell
sont les
to see
gènes qui sont
what
exprimés dans
genes
ces
are
cellules en
expressed
réponse au stress.
in these cells
Et son
as a response
objectif
to
est de
stress. And her
comparer les
objective
réponses au
was to
stress
compare
mitochondrial
the
des neurones
mitochondrial stress
dopaminergiques par
responses in
rapport aux autres
dopaminergic
sous-types
neurons
cellulaires
versus
et
other
voir
cell
si ces réponses sont
subtypes,
différentes en
and to
contexte de
see
déficience en Parkin.
if the alterations in
Alors,
these
je vais vous
responses
montrer
in the
ces
context
résultats
of Parkin
vraiment
deficiencies. So I'm going to show
préliminaires,
you these
c'est-à-dire
preliminary
que,
results.
pas plus tard que la semaine dernière.
They're from last week.
Et donc, voilà, en regardant tout ça, eh
Looking
bien,
at all of this,
donc, sur la gauche, vous avez des
on the
images
left,
de coupes
you see
d'organoïdes. On les
images
a
of
coupés
cross
et
sections
puis
of
on est
organoids.
capable de marquer
And
les
so we're
types
able to
cellulaires.
mark
C'est-à-dire
the
qu'on
cell
va
types.
marquer en haut en
We
rouge, ce
can
sont les
mark on the red
progéniteurs des neurones
at the top,
dopaminergiques,
you have
c'est-à-dire les cellules qui
neuron
vont donner des neurones
progenitors,
dopaminergiques majeurs.
dopaminergic
Au milieu, c'est
neuron
des neurones
progenitors. In the middle, you
GABAergiques
have
et puis en bas, des
GABAergic
astrocytes. Ce
neurons,
sont des
and
cellules
at the bottom, you have
épicales comme le
astrocytes.
cerveau. Et on
These are
voit
other
en
brain
réalité
type
que
cells. In reality,
les
what we
contrôles
observe
et les
is
organoïdes
that the
qui n'ont pas de Parkin,
controls
c'est
and
marqué K,
organoids that don't have
eh
Parkinson's
bien, ils ont des compositions cellulaires
have
différentes. Vous voyez qu'il
different
n'y a pas
cellular
la même
composition
organisation du
of
marquage et
ventral
même des
membrane
fois
organoids in the absence of
beaucoup
Parkin,
moins de marquage, comme pour les astrocytes.
much less marking.
Ensuite, elle a fait, et elle est encore
And
en train
then
d'analyser, parce que c'est beaucoup
she's
de travail,
continuing her analysis because it's a
ce
lot of
qu'on
work,
appelle donc du séquençage en cellules uniques, où on regarde
a
dans
single
chaque
cell
cellule l'expression de
sequencing to
tous
see
les
the
gènes.
expression in each cell, the expression
On
of
le
each gene.
représente par les schémas que vous
So this
voyez,
is
où en fait
represented
chaque
by
point
this
est une
diagram you
cellule,
see here.
d'accord?
Each point is a
Et
cell.
en regardant tout
And
ce
by
qui est
looking
exprimé dans ces cellules,
at
on est
everything that's
capable de
expressed in those
dire,
cells, we
de
can
donner une
actually
identité à
say,
ces cellules.
give the
Ça veut dire
identity to the
quel
cells to say
type cellulaire
such
on
a
a.
subtype we
Ainsi, ils
have.
sont représentés par couleur.
So they're
Donc, vous
represented by
voyez en rouge,
colors
par
that
exemple, c'est
reflect
marqué
that. The red
PDA,
ones,
ce
it's
sont les
PDA.
progéniteurs
These are
dopaminergiques. Et juste à
progenitors,
côté, en vert, un peu en dessous, les
dopaminergic
neurones dopaminergiques.
progenitors.
Et qu'est-ce que vous voyez? Ben, vous voyez
And so what do you
que
see?
dans les organoïdes qui n'ont
You
pas
have
de
in the organoids that do
Parkin,
not have
en bas,
Parkin,
il y a
at the
plus de
bottom,
progéniteurs
there are more
dopaminergiques que dans
dopaminergic
les contrôles.
progenitors in the
Ok.
controls.
Et de même, on a des différences au niveau des sous-types
Similarly, we have
GABAergiques
differences
ou
in
des
level
astrocytes où il y en a
of the
moins. Ainsi, ça
subtypes,
montre
where there are fewer,
qu'on a
which
une
demonstrates
altération de la
that we have
composition
an
cellulaire des
alteration of the
organoïdes
cellular
des
composition of the
mésacéphales
organoids
en
in
absence
the
de Parkin.
midbrain
Ce
organoids
qui veut
in the
dire que
absence of
Parkin pourrait
Parkin. In
jouer un
other words,
rôle dans les
Parkin
processus
could play
très
a role
précoces
in the
de
very
développement.
upstream
Bien sûr, c'est à confirmer
developmental
avec des
processes,
expérimental et
confirmed
à
obviously with
contre-temps
different
de culture. Par
additional
contre, en plus particulier
experiments
parce
in
que
different
ces
types of
différences sont
cultures.
compensées avec le
These
temps.
differences may be compensated over
Et
time.
puis, pour finir dans les résultats,
So
ce
finish
que je vous ai
with
montré, c'est
the
en
results,
condition
what I
basale.
showed you,
En
it was
termes
a
de
basal
réponse
condition.
au stress des
The stress
mitochondries,
response
eh
of
bien, on a pu voir que le
mitochondria,
stress chronique
we're seeing that
accentue les
chronic stress
altérations cellulaires liées à l'absence
increases
de Parkin,
cellular
c'est-à-dire
alterations in the
les
absence of Parkinson's.
organoïdes qui n'ont pas
So the
de
organoids
Parkin
that have no
vont avoir, par
Parkin
exemple, encore plus de
will have
progéniteurs dopaminergiques
more
que les contrôles
dopaminergic
et
progenitors
moins
than the
de
others, and less
neurones dopaminergiques. En
dopaminergic.
revanche, en réponse, quand on regarde l'ensemble
But
des réponses,
when you look at the
on voit aussi qu'il y a
whole set of
une
the responses, we
différence
see that
dans
there's
le
a difference
traitement.
in the treatment.
En fait, je vais être plus clair, chez les
To be
contrôles comme chez les
in the
organoïdes qui n'ont pas de
controls
Parkin,
and in
le
the
stress
ones that
entraîne
have
un
no
fonctionnement
Parkin,
un peu
stress brings
plus
about
important des
a more
mitochondries.
important
C'est ce que vous voyez
effect
en
on the
particulier avec les
mitochondria. You
flèches
can see that
vertes.
presented by
Et
the
chez les
green
contrôles,
arrows.
eh ben, il va
And
y
in
avoir
controls,
des
there's
processus de
also going to
protection
be a
des
protective
protéines.
effect
C'est-à-dire
of
que si
proteins.
par le stress, les
In other
protéines ne
words,
sont pas
if
très
by
bien
stress the
repliées ou
proteins
ne
are
fonctionnent
not
pas très bien,
well
on a
folded or whatever,
des éléments qui viennent
we
là
have
pour les remettre en place et
something
bien les replier. Ça, c'est ce
that
qui se passe dans les contrôles. Eh
refolds
bien,
them
dans
properly.
les organoïdes sans Parkin,
So
eh bien,
in
il y
the
a non seulement ce
organoids without
processus n'est
Parkin,
pas là, mais en fait, il est
the
même
process is not there,
désactivé. Il
but
y a
it's
donc
even deactivated.
une réponse au stress
So
qui
one
est
response to
dysfonctionnelle.
stress, which is
Il nous
dysfunctional.
reste maintenant
Now, what remains to be done is to compare these responses to stress with responses in other cell types. I'm going to conclude by explaining that this organoid approach to studying the molecular mechanisms involved in Parkinson's disease is using human cells. So there's possibility of working with patient cells. It can also be used as a test platform. We can assess different types of molecules for different therapeutic approaches and confirm the genes identified by genetics. I showed you the example of Christian, who's working on a stress response, but we also have Elena in the laboratory who's been working on neuroinflammation in Parkinson's disease, Elena Winterberg. You can see on the video. We also have Pauline Lachal, who has a poster here if you'd like to see. She's working on the establishment and functioning of neural networks, the assembloids, in presence or in absence of Parkinson's. And we can also work, as Annelle is working, to work chimeric organoids to validate the genes associated with Parkinson's disease. I hope I stayed in the time. In any case, thank you so much for your attention. I'd like to thank my team. It's a kind of a big team. There are a lot of us. And the organoid poll, and you have quite a few members here today. Thank you.
Merci beaucoup. Vous m'entendez toujours? Oui, merci beaucoup. Donc, vous comprenez, là, on est vraiment dans de la recherche très fondamentale, dans les mécanismes.
This is really fundamental research. It's not always easy to explain, but I think it's important to realize that this research does exist. It's necessary so that we can do some research on clinical molecules. So thank you. It was very important to talk about these questions, even though it's a bit more complicated to explain. We have a few questions that we've prepared. Perhaps we could have the first question, please. We talked about organoids. Why do researchers use organoids in studying Parkinson's disease? To model small human brains to replace MRI or to look so that it looks pretty in the labs. Okay, we're trying to relax you guys. A little transition here.
Je pense qu'il y a des gens qui nous font quand même des clins d'œil, comme nous on le fait. Donc voilà, donc n'hésitez pas, après il y aura tout le temps de poser des questions aussi sur ces
You can always ask questions on the organoids.
Ce ne sont pas des vrais mini-cerveaux, comme vous l'avez dit.
They're not mini brains, as you may have realized, but we could talk about that afterwards. Thank you.
Voilà, on n'a pas de nuage de mots là, on n'a pas eu d'inspiration.
We don't have a word cloud.
On ne savait pas comment appeler un organoïde, donc on s'est arrêté.
We don't know what to call an organoid, so we're going to move on to our next speaker after the jingle.
Vous allez adorer cette musique.
Bonjour à tous, le micro marche.
Hello, everybody. Apparently the microphone is working. Right, I'm going to take a little step back and look at the whole animal. Not so much the motor symptoms of Parkinson's disease, but rather the non-motor symptoms, especially the pain syndrome, which may appear in Parkinson's disease and which we've been trying to model in animals. And I'd like to show you work that was partially funded by France Parkinson NFSC regarding the role played by another monoamine that we have talked about dopamine so far, but now we're going to talk about serotonin. This is a rather general scheme that we use when we look at the Parkinson's disease evolution. Here we have all the motor symptoms in the disease that most of you are familiar with. And also at the bottom, in the lighter green shade, we have the non-motor syndrome, which also can be rather debilitating and can appear very early on in the disease. or even sometimes before the disease is diagnosed. Pain is one of those symptoms, non-motor symptoms, very debilitating motor symptoms, which may appear in early stages of Parkinson's disease. Pain in Parkinson's disease, just like any other pain suffered by patients of any kind, has several dimensions. There are syndromes associated with the posture defects and musculoskeletal disorders, but there are also kinds of pain that are called central and are difficult to account for with the motor disease and are difficult to treat. It says here that depending on the studies, there may be up to 80 or even 85% of patients suffering from pain. If we want to draw up a table with the percentage of pain-related symptoms, most of them are motor issues, posture-related issues, musculoskeletal disorders, but there are some symptoms which are either neuropathic, they or have to do with radicular neuropathic disorders, and in some cases, up to 30% of central pains which have nothing to do with motor symptoms. And unfortunately, central pain is the most difficult kind of pain to treat. So far, with regard to pain treatment in Parkinson's disease, for all the motor symptoms related pains, we have medication for that kind of problem. We also have interventional therapies such as the brain stimulation, and it may sometimes improve pain, especially musculoskeletal pain. But there are also non-pharmacological therapies, yoga, acupuncture. But in spite of all these efforts, unfortunately, some patients are still in pain and nothing can relieve their pain. In the lab, we've been trying to come up with a model, understand what is really going on and why there are types of pain that cannot be relieved, although we improve the motor symptoms. This graph shows you that chronic pain means daily pain that will last for more than 3 months consecutively. And this is a definition that can be used for any pain-related disorder. Pain will appear after a certain threshold of stimulation, mechanical stimulation. And when you start stimulating up at a certain threshold, you activate the pain fibers, and the more you activate, the greater the pain. And if pain becomes chronic, the curve will be totally offset, and the pain will start at very low stimulations, allodynia, or pain will appear for stimulation, which is normally painful, but the response is exacerbated. Just a quick reminder on how pain is perceived by the brain. There are so-called nociceptive pathways that will carry information which is potentially dangerous for the body and will be perceived as pain. We have pain receptors. Here they are in English, but heat, cold, pressure, and the information reaches the dorsal horn of the medulla, and then we have of the spine cord, and then the information will be sent to the brain and will be perceived by the brain after it has traveled through the spine cord. But it doesn't stop there. The brain can send information back to the spine cord to control pain, and this is what we call endogenous control. All the curves I showed you earlier, for each individual, there may be a variation in the threshold that triggers the disease, the pain, sorry, a variation in the maximum intensity of the pain. And this is something you may have experienced in your life, what we call endogenous control. Endogenous control can be manipulated. Here we have a quote regarding combat zones when there is no correlation between the size of the wound and the pain perceived, but also stress-induced analgesia. For instance, boxers during a fight who will not feel the pain, and after the fight, yes. And there is also the so-called gait theory. You hit yourself with pains, and then you rub the area and you activate your endogenous control. And finally, we have also this piece from a very famous French film where Josiane Balasco has, the actress, has dislocated her shoulder, and they try to attract her attention so she will not feel the pain anymore. And there are descending controls as well coming from a number of brain structures. They reach an area where yellow, red, and green areas can be found in the brainstem. There is dopamine controlling the pain, and I am very interested by serotonin neurons in the spine cord, in the cerebral stem, in the brainstem. We need an animal model, so we will cause dopaminergic lesions, very brutal, reproducing advanced stages of the disease. In green, bright green, we have the dopaminergic fibers, and when you injure them unilaterally, you have one side where there is no more dopamine at all. Now, when we do this, we reproduce motor alterations. I'm not going to describe them, but the animal will not move around so much in the maze, will start turning around because it's unilateral. So the animal will only turn one way and not the other way. And the animal, if you test the sensitivity to pain, here we see filaments, and we apply them on the animal's paw, and we look at how much force is needed for the animal to withdraw the paw. And here we have a sham control. It takes 1 gram for the animal to withdraw the paw. And in Parkinsonian animals, we need 0.3, 0.4 grams. That will be enough for the animal to withdraw the paw. So we went on to look at what happens in the serotonin neurons in the brainstem controlling pain. First of all, we see dopaminergic fibers coming from the mesencephalon contacting the serotonin neurons. Yellow contacts, beautiful synapses. There is a connection, obviously, between those areas. And in the Parkinson model, we see a decrease in the number of dopaminergic fibers in the area. So we went in to look at whether with this decrease, there was also a disruption in serotonin neurons. We tried to use transgenic mice, a very special model, I will explain later, to spot serotonin neurons. And we used PR, which is a cellular activity marker. The greater the number of PRs, the greater the activity. And with serotonin neurons in our Parkinson model, we see a decrease in the activity of serotonin neurons. And if we look in the spinal cord, we see an increase in the level of serotonin in the spinal cord. And it seems like in our model, serotonin neurons are hyperactivated. So we tried to inhibit them to see whether we could improve the pain-related syndromes. So we used a special kind of transgenic mouse, and we have a way to specifically inhibit those neurons. And when we do that, you see that the threshold to elicit a paw response, the threshold is not the normal threshold. So by inhibiting the serotonin neurons, we can improve the pain responses in our Parkinson mouse model. The idea here is to find a way for future therapies, and we tried to look into pharmacology and see what was actually involved in the hypersensitivity, especially serotonin receptors in the spinal cord. And I'm going backwards, but in a human being, it's difficult to inject auxins, to place optic fibers, switch on the light, and see what happens. Maybe we can do it one day, but I don't think we will. And pharmacology is therefore a better way. So we tried to look at antagonist receptors for serotonin to try and see if we could mimic the effects by inhibiting the response to serotonin. The antagonist is going to block the effect of serotonin on the receptor. If we use an antagonist with a very complicated name that stops the 5-HT2D2A receptors, we have a slight increase, but it's very limited compared with what happens in non-painful mice. If we place the 5-HT3 antagonist, another slight improved, but not enough yet, and both together, if we use both together, then we have a threshold that matches the one we had found in the control mice. Or it seems that if we combine the FAVE-HT2A and FAVE-HT23 receptors together, we can improve the painful syndrome, at least this one in Parkinson's disease. So in summary, there is a preclinical model for Parkinson's disease obtained by a lesion on the dopamine neurons. We have a loss of dopaminergic fibers in the brainstem, increasing the serotonin neurons activation, neurons that control the transmission of nociceptive perception. And by doing this, we increase the level of serotonin in the spinal cord, which in turn will be responsible for hyperactivity in the nociceptive systems, generating hypersensitivity. And we can therefore control this with a 5-HT2A and 3 receptors. The work was performed by my former PhD student and was published this year, so it's very recent. So that was the story, and I would like to thank all the people who are working with me in the team in Bordeaux, especially Zoé Grivet, who is our PhD student, and she did her PhD in 2023. And in 2025 now, it shows you how long it takes for research to be published, but I thank you for your attention. Well, thank you very much. It's important that we show we're not just looking at motor symptoms, but also non-motor symptoms, because they're just as important. And it's vital that we understand the preclinical fundamental research to understand the mechanisms, because there are things that we cannot do, obviously, in human subjects, and we have to use experimental models. Maybe in a few years from now, we will have a double antagonist, which will really allow us to relieve the pain. But again, we first need to understand the mechanisms so that we can reach the clinical research stage. Now, this was a 15-minute presentation, obviously, but it summarizes years of research. It's like organoid. There was a time when organoids, we didn't know what we could do with organoids, and an organoid is all very well, but it takes months to obtain something that can be used for research. This kind of research is very important. It gives us an idea of how long it takes and why it takes so long to move forward with research. Well, thank you very much again for this presentation. We'll move on to questions. And the researchers have not been informed of the questions beforehand, so they discover them at the same time as you discover the questions. So if you listened well, it will be easy. What percentage of patients suffering from Parkinson's disease report pain? Again, what is the percentage of patients suffering from Parkinson's disease who report pain? 85%. Yes, it is 85%. The percentage that was shown in one of the slides. And I think we do have another question. Now we have a cloud again of words. So this is a question in one word. Where do you feel pain? So if you suffer from Parkinson's disease, we would like to know where you feel pain in one word, because we know that Parkinson's disease symptoms may vary a lot from one patient to another, and maybe you feel pain in very different places in your body. I would like to take a photograph and show this to the Parkinson Center in Bordeaux, because in Bordeaux, Parkinson patients are not in any pain. I don't know why. That's very different. So don't hesitate. Here we're talking about preclinical research, but it's very important when you have pain to talk to your doctor, your neurologist, because maybe there is a solution. Maybe something can be done to alleviate the pain. Don't think it's just something that you have to endure. You can talk to your doctor. Something can be done. Okay, we'll give you a few more seconds because I see people writing. And also, we can see the word clouds on the replay. I think that we're recording this event, and so it can be watched afterwards on the YouTube channel, and the word cloud will be there. I would like to add that sometimes, or very often, we don't try to assess the pain in most of the Parkinson expert centers. It doesn't mean, obviously, that people are not in any pain and bother. It's just because the pain is not being tested for and assessed. We look at the motor symptoms more than the painful symptoms. Well, France Parkinson supports the development of a new scale to assess pain in the Toulouse Center. We are understanding that the scales to assess pain are sometimes contradictory. And I think it would warrant a publication. There should be a publication very soon. There is going to be a questionnaire to really try and detect pain in Parkinson patients. And it's currently being validated not only in Toulouse, but all the expert centers in France participating in the project. Okay, I think we can stop the word cloud now, and we can move on to the fourth and last presentation with Dr. Louise-Laure Mariani. The jingle comes first.
La parole est à vous.
Donc, si vous avez encore un peu d'énergie pour la
If you have a
suite,
little energy left for what is to follow, I'd like to thank the whole France Parkinson's organization who have invited us to talk about what we do, talk about it together. I'm going to be talking about how do you prevent the undesirable effects of medication that's used, and the study that was financed by France Parkinson. I can base my remarks on what was already introduced by different people here. So thank you for having prepared the terrain for me. So we've talked about the disease. It's neurodegenerative. There's a clinical triad with a gradual appearance. There's a trembling, bradykinesia, and there's a rigidity which can bring about contraction or cramps. And now these motor symptoms, not everyone has all the symptoms, but are due to neurotransmitters, not in the brain. These neurons are actually in the transcerebral, it's in the black matter. This is an idiopathic because there's not one single you can identify cause. There are many different causes that participate in the appearance of this disease. So the suffering of dopaminergic neurons, I can't point it. You see the cross section of the brain. You see on the left. We have a non-pathological situation with a structure which appears as being black because it contains neurominanin, like for the skin. But it's called, and the usual thing, we have a transmission normal with dopamine, which is going to transmit a message to the other parts of the brain. On the right, we have someone with Parkinson's disease. We have these neurons in link with the aggregation of proteins, little packages of neurons that are going to cause the suffering. Let's look at that little. So there's a lack of transmission of these dopamine received in the brain, which is going to result in motor symptoms that we just spoke about. Compared to other neurological diseases, there's a lot of medical treatments that are available, which are mainly symptomatic treatments with a view to improving the symptoms. Now, the global strategy is to replace the missing dopamine. For example, we could use a precursor as a dopamine, such as levodopa, or that will mimic the action of dopamine, such as dopaminergic agonists. So these are prescribed, and these medications are quite effective on motor symptoms. That depends on the patient, obviously. But the dopaminergic balance is something that's quite fragile to monitor. You have to keep an eye on it. Physiological, we're in a state of balance. We try to maintain that balance of amount of dopamine, even if it oscillates a bit. But in Parkinson's disease, you have a hyperdopaminergia, which brings about motor symptoms, as we've already spoken about, anxiety, apathetic, depression, and a problem of planning as well. So when you give a treatment, we're going to be improving the level, but sometime we're going to do it. So you're going to be over supplying dopamine, which can bring about the opposite response. And sometimes euphoria, hypomania, craving, or imperious besoin, or it could be addictive, as was already spoken about by Christian. So dopaminergic agonists are very effective, but they have some undesirable side effects with a hypersensitivity. It can be behavioral that are hard to control. It can impact the quality of life. You see the type of behavior can be impulsive with a hypersexuality, hyperphagia, with attachment to sweet or fat foods, or it could be compulsive buying. So it could have a financial impact as well, or people that like to play gambling games. This doesn't happen from one day to the next, but these are undesirable side effects that you have identified, and we try to make sure and watch for these the appearance of these types of behaviors so it doesn't impact negatively their lives. There are a bit more pleasant aspects. One patient was a patient. He totally changed his context with a much more neutral context, and with higher doses, the context was a bit more sexual. Here's an artist who reported that when he was under treatment, he was the most creative thing in his life. But it was accompanied by all-nighters and different types of symptoms that with his creativity it did bring about creativity, but it was hyperstimulation as well. This is another type of behavior, is punting, repetitive behavior with no purpose. It's kind of a stereotype. Here's an example of someone who's trying to guess the horse race results. But it can be when you take things apart and put them together again, or you're knitting, you knit and then you unknit. It is repetitive. So these symptoms, the intensity can vary, but they're quite frequent because after 5 years, half of patients have these symptoms. So they're really under careful watch on the part of patients and the physicians, and that's why I'm talking about it today, because if we have these symptoms, you have to let the doctor know. We don't necessarily say we're not really sure it's due to medication, so it's important that they know this can happen. So now you can have the situation on the left where you have the therapeutic effect, the patient's happy, they're feeling quite well, or in the middle, no effect or not enough effect. So we might have to increase the dosage to have the right effect. And on the right, the adverse effect. And the purpose of the EPI-TCI project is to understand the biological mechanisms that are associated with these undesirable effects and why some patients have these effects and others don't, and to do this to develop therapeutic strategies as a consequence of that. So the project as such, we worked with mice in Parkinsonian or non-Parkinson's groups, and we gave 3 weeks of treatment, either paraminergic or non-active, placebo, if you like. And then we evaluated the behavioral aspects, either by placing a mouse on a platform that they could explore freely, or a task where subsequent to a light, they would do some tasks to win a reward. And once we assess these behavioral effects in the different groups, with or without Parkinson's, with or without treatment, we went to see in the various areas of the brain what modifications of gene expressions occurred in conjunction with these behaviors. So this is the transcriptomic. So these are the first results. That's where it would be good to have a laser pointer. This is the platform. On the left, you have, in the first chart, the distance run. We put everybody in the same level. All of the mice were behaving the same way, basically with no treatment. They just go walking around. And then the second chart, in distance, you have in purple the Parkinsonian mice and gray without Parkinson's. So you can see the Parkin chaos, as Hélène was speaking about. But you can show you, we also did this in another model, hydroxytopamine treatment, and they introduced different models. So we have similar results, whatever the model we used. And so in purple, you have the ones with Parkinson's, and gray, not Parkinson's. And the full ones are treated by non-active, and empty, they're treated by the agonists. And whether you're in a Parkinson's group or not, we have an increase of the amount walked. And the intrusion outside of platforms, where the mouse spontaneously stays in the middle to be protective. They're not going to go all over the place. But when you look at the intrusions on the edges, that is still the same. The mice treated with the agonist had a tendency to go around to the edges of the platform without any attention to risk. Sometimes they would even jump. And that's the last chart. We can see that mice treated with agonists had a tendency to jump from the platform and be more impulsive. So this leads towards impulsivity. The distance walked and falls as well. And another test. This time we had to accomplish a task to get a reward. So the agonist treatment induced a slowing down of the performance of the task, the latency of response, the time to respond to the light and go get the reward. With mice that were treated by agonists, the symbol is empty, you can see the latency is always below. They're always slower to accomplish the task. In the same way, sometimes they're so slow on the chart on the right that they're might not even respond. They might wait too long and not do the task at all, not get their reward. And they have less premature responses than the untreated mice. You can see that medication treatment is anotypical. It's very consistent, whatever the model, in different animals. And so we sought to find the biological substrate that were affected by these different treatments. So a concentration here, we have what we call a volcano graphic. It looks like an eruption. On the right, in the blue lines, you have genes that are increased. And we found in orange, the expressions were up significantly. 42 increased significantly of these genes in mice that were treated with agonists. And on the left, you have the blue, the expression was diminished. So they're diminished significantly. Here you have 31 genes that were diminished. So in total, we have 73 genes where there's a change in expression induced by this treatment, which are associated by behavioral results. So we looked at in the various subparts of the brain, and this was introduced by Christophe Boudez, in the structures that could be involved in addictive behavior, for example, prefrontal cortex, and the dorsal, which is that one structure that is particularly receives less dopamine with Parkinson's. That was a bit individualized because the signal is more disturbed than in other structures. And we realized that there's one single gene that was in common in all of these. So it was more important than all of the rest in terms of biological modification. The structure that had the most modified gene, 40, was a structure in the dorsal area that had the most effect on Parkinson's. Then we looked at the profile of global expression, and here This is a heat map in English. In each line, you have a gene that is represented, and each column is a mouse. And what we realize is that when you look at the global profile of genes, you can separate two populations of mice. In purple, those who are treated by the dopaminergic, and in gray, you have non-active. So we're able, with the profile of a genetic expression, you can see the different behavior and effect on treated and untreated animals. So in patients, in an international cohort called PPMI, 549 patients out of 1,900 visits, and we realized that amongst the genes that we found modified in the mouse, there were genes that were involved in the same pathways as in the mice. We observed that they're also modified in patients. So all of this seems to reinforce that the pathways we saw as modified were implicated more credibly in the modification with patients under treatment. So what is to follow? What is the lesson? We've discovered behavioral modifications that are induced by agonists, whatever the situation, and that are associated with specific modification of gene expression. The next steps would be at the bottom here. In cerebral tissue, we will be separating various populations of neurons that are involved differently in motricity or in addiction. And we're going to be then looking at the genetic expression specific to each subcategory of neurons to better understand the imbalance generated. And then we can perhaps better balance those aspects. In the future, in the idea of personalized medication, because we're not robots, every human being is different. To move from the situation I described earlier, which is on the left, in other words, a prescription, and you can see the therapeutic effect or absence of effect or bad side effects, or we hopefully will be able to individualize each treatment so that everyone could have a good efficacy without side effects. We have a project that's supported by the ministry called Prevent ICD, which is an application based on artificial intelligence that was developed at the lab, and the clinic is going to start in 2026 for people who are under this agonist type treatment to adapt the treatment to the right here as a function of the characteristics of each individual so that we can optimize those as best we may. Thank you. I'd like to thank all the members of my team. We work really hand in hand on Parkinson's. This is a group that I've been working with very closely. All members of the team, the students and masters, Corentin and Kate, who participated in the project, financed by France Parkinson, for the aspects having to do with patients, and two others that were data scientists and who obviously we use their expertise from a data management point of view. We didn't go on a weekend. For those of you who you'll recognize, this is the Parc de la Hauteur, which is near the Institut du Cerveau. So this is everybody that participated in this research on this particular topic.
Merci beaucoup. Je pense que vous nous avez donné aussi un aperçu de ce que peut être la recherche translationnelle.
So this is translational research, how you find experimental models you can then compare with human samples and have the same type of modification. And you start with experimental and you move on to specific applications in the human. And this is a very particular theme. We started a big survey on the national level to ascertain the way people understand it. We're going to have the results of that survey soon. It'll be interesting. And we have prepared some questions here for the room. And then we'll move on to what side effects can be associated with dopaminergic treatments. Heart difficulty, controlling your impulses, no effect, hair loss, or night vision improved. So what are the undesirable effects that can be associated with dopaminergic treatments.
Donc, on a quand même des chutes de cheveux.
We have hair loss.
On va peut-être identifier les personnes qui ont
Maybe we're going to identify the people that answered in that way.
On va passer à la dernière question pour la salle.
Let's move on to the last question. It's a word cloud, I think. A difficult question. If the side effects were a character in a film, what film would it be? Or what character would it be? The Joker?
C'est étonnant, on était assez sûr que ça allait apparaître.
We were sure that was going to pop up on the screen. Terminator, Dracula, Woody Allen, Flash, Scarface, Mad Max. So we're going to let this run, and then we're going to move to questions in the room. And for people online, you have a chat that's dedicated to that. We are also translating online. We might have people that are following us in English. You can ask your question in English. If you wish, we can translate those into French. For questions that are coming up, the first question from the floor is always the hardest one. Once we started out with the questions, there are no bad questions. Sometimes there are whole areas of research that started thanks to questions from the room. So that's not without interest. All questions are interesting. So please don't hold back and get our researchers out of their zone of comfort. Take advantage of their presence here to put forward your ideas. Are you ready to answer questions from the floor?
Someone had a question right from the beginning, so we will start with this gentleman. Please raise your hand. We have 3 roaming microphones. Please wait for the microphone. I thank the speakers for their very interesting presentations. I have a question regarding therapy. We have experienced 20 years of failure in gene therapy of neurodegenerative diseases. Well, I'm exaggerating, but successes were few and far apart, except for a few diseases. Huntington's disease is a very good example, which was considered to be the one that could be treated with gene therapy. But adenovirus with wild type genes were a failure. But more recently, a biotech company showed that by performing gene therapy with adenoviruses without the wild type gene, but rather containing an RNA population called microRNA, regulator RNA, we achieved some very promising results. In Parkinson's disease, 1, are there any trials looking into the role of microRNA? And 2, are there any trials that are thinking of using microRNAs for a therapeutic solution? Maybe I will answer by saying that there are studies on trials with microRNAs in Parkinson's disease. We supported one project at the Institut du Cerveau a few years ago, and then I'll allow the researchers to answer. Sébastien Cancela, Dr. Rabassa, the Institut du Cerveau, the Brain Institute, they both worked on microRNAs. Please, you answer before I say anything stupid. At the Brain Institute, we work on another form of microRNA, of RNAs, long RNAs, very different. For microRNAs, a lot has been done for all kinds of neurodegenerative diseases and other brain conditions and situations in which there are dysregulations taking place. I wasn't aware of this trial that you just referred to, but the idea is the following. Those molecules can regulate gene expression, and technically they could correct, I guess, the defects that are found in the disease. It's It's a new approach. It's been around for quite some time. We are working on microRNAs for many neurodegenerative diseases. It's an expanding field of research. But again, I know long RNAs better than microRNAs. Well, generally speaking, because maybe this is not very clear for everybody, there is a new therapeutic area being studied, but not just Huntington's disease, SLA, Huntington's disease, and not only microRNAs, but interference RNAs, other RNAs. The idea is when in the disease, one molecule is being overexpressed, we will stop its expression. And then in other diseases where a molecule is missing, we will try to restore the expression. It's been done for myopathies. It's been done for some very serious diseases, infantile spinal amyotrophy, children who die within 1 year, and the new therapeutic solution can be applied when the child is very young, and screening must happen at birth, but there is a solution. So there is a lot of work being done in neurosciences on RNAs. It's very promising, and there are trials on multi-systematized atrophy. There is work in progress with intrathecal injections of RNAs that modulate the expression of a protein, in this case, fascinuclein. Okay, another question here in the front row. Thank you. As you said, no stupid questions. I have 2 questions. May I ask? Well, ask one first. On the experience on rats and addiction, why did you not come up with a protocol in which rats have no interaction? Well, we perform conventional tests in conditions where they have no interactions. That's the gold standard, that's the reference. We always do that. We test them in situations with no interaction. And for the last manipulation I showed you, because they push the lever to open the door, experiments have been conducted where behind the door, rather than placing a rat, we place an object to see whether the rats were as interested in an object as in a cage mate. So I'm not revealing anything new. Obviously, the rats are not so interested because an unmoving object is definitely going to make them feel curious the first time, but not the second time. Very quickly, they'll lose interest. One more question, and then we will have a look at the questions online. I think alpha-synucleic acid was mentioned for dopaminergic neuron degeneration, and I heard you talk about mitochondria degeneration. Does it mean the target has changed? I'm sorry, I don't. Okay. Mitochondria are elements contained within the cells. If you take a cell and you consider it as like a small town, and the mitochondria will be the factory where they produce energy. And all the cells have this kind of little factory inside. And mitochondria do not necessarily degenerate, but sometimes they may work in a less efficient way in the neurons. And when all the cells are affected, like in Parkinson's disease, we believe that in dopaminergic neurons, mostly, the protection mechanisms that have been implemented to counteract the mitochondria degeneration are not quite as efficient. And that's why dopaminergic neurons die. So we believe that the protective responses related to mitochondria dysfunctioning are not functioning well in the dopaminergic neurons. Have I been clear? Well, we're talking about something that happens within the cell and not a different cell. Mitochondria can be found in any cells. I'd like to answer a few questions online because we have quite a few. There is a question for Dr. Cheval, a more technical question. How long can we keep organoids in culture?
Alors, c'est une très bonne question.
Very good question.
On pourrait presque dire le temps qu'on veut.
Almost as long as you wish. All the PhD students in the room will shed a tear. No, we seriously, we keep them for 2, 3, maybe 5, 6 months. We cultivate them and we collect them after a couple of months, sometimes 5, 6, depending on the biological issue that we're working on. In the project that I showed you at the end of my presentation, we assemble them, we make assembloids, and we allow the connections to happen between the various areas. And it's Pauline Lachal's project. She looks after 3 months and then after 6 months. And I think she even went further than that. And just to take time in consideration, how long does it take to obtain an organoid? When you start cultivating the very first cells, how long does it take to obtain an organoid? Well, I think we can start talking about an organoid after a few weeks, but it's a very immature organoid. Then it will continue growing, developing. I mean, we want to mimic embryonic development, basically. And then we increase the maturation with time. And in order to obtain mature dopaminergic neurons, projections that work, it takes longer. We wait longer. But that is because we want to obtain organoids. Then there are the experiments we're going to conduct on the organoids once we have obtained them. And that takes time. So one single experiment could last maybe 9 months, 12 months altogether. I have a question online on pain. Another question online. A question on pain. Serotonin receptor inhibition in the spinal cord, could that induce secondary undesired effects in man? Of course. Of course. The preclinical research results do not let us believe there will be in the near future a therapy based on the antagonists. Serotonin also has an impact on the motor area. There are receptors that play a role in the motor control, but we have ideas that we could look into to understand how we could maybe come up with a better treatment in the near future or in the future. But obviously, we still need to go over a certain threshold. If you'll allow me, I'd like to push the question further. You talked about molecules that have been tested on their own and also in combination. Can we imagine that by combining them, maybe we could apply a lower dose? And by applying a lower dose, we could limit the undesired effects? Well, of course, we can work on lower doses, decrease the doses, but we can also develop new molecules which possibly might target more specifically those 2 receptors only. But again, that means we have to perform biochemical research, biology research, but we have new avenues we need to explore. Right now, we have this result. For what it's worth, it has shown that he can improve pain-related syndrome. There are also thermal response issues that I have not had time to mention. It's promising, but we're still in the preclinical phases, and we need to still develop the hypothesis to have a more specific treatment for man. One more question in the audience. Hello, I had a question regarding agonists and unwanted effects. Have you used in this study the mode of administration, oral administration or pump administration? In this study that I mentioned, we did not use oral administration. Well, we did a test that I have not mentioned because we wanted to compare, I mean, we compared attraction for glucose, and we compared the glucose and the agonist. There seems to be no preference for agonists, but that's the oral administration. In the study, we did not perform oral administration. We gave the animals a chronic therapy one dose daily, which is close to what happens in patients who take the medication orally, but we do not use pumps, if that's your question. Some unwanted effects are not quite so present if we use the pump rather than the oral administration. Apomorphine is the molecule, because it's a continuous administration with a pump. And the more continuous the administration, the lower the stress, and therefore the lower the unwanted effects. Maybe a patch would give a continuous release that would be better than oral administration with immediate release, but it's pharmacokinetics here, and the way the administration can play a role. We tried to put ourselves in the situation that was as close as possible to the study I showed you that showed 50% after 5 years of treatment. Another question from the audience? Thank you for the presentation. Please put the microphone close to your mouth. Can you hear me? Not so well. Okay. I have a question, basic question, not very technical. I'm wondering, knowing the adverse events is not recent. I mean, we've known about them for a while. So why, when a doctor prescribes the treatment, are we not warned against unwanted adverse events? I am playing an active role in my therapy, and if you explain to me that there are unwanted effects, and maybe I will act accordingly. And the underlying question is, if I don't take the therapy that will act on my symptoms, does it mean that the disease will evolve more quickly, or will it still evolve at the same pace? So are we talking, on one hand, the symptoms are not present, and on the other hand, does it have an impact on the speed at which the disease will evolve? Because we have to be active players in our therapy. Absolutely, you are actively taking part in your therapy. I mean, this is the kind of conversation we have all the time with our patients when we introduce the treatment and during the follow-up. But there are several questions here. First question, is the therapy playing a role in the speed of evolution of the disease? Well, the therapy improves the symptoms, but it doesn't change the course of the disease. And that's why we continue research. And in my clinical center where I work, we have conducted many trials on neuroprotector therapies, a different approach from the one I introduced today. And the therapies we offer nowadays, what you can buy in nowadays is something that will treat the symptoms and not change the course of the disease. There's only one for which there was one trial showing that maybe it plays a small role in the evolution of the disease, but not enough for us to stop research. And so taking those drugs is mostly to improve your life, your quality of life. And if you, I can show you pictures who were taking earlier in the century, when before those drugs were discovered, when the Parkinsonian patients would be hospitalized in psychiatric wards, they were in a very bad, poor condition, and not taking the medication, relieving the symptoms was very bad for their quality of life. Second question, unwanted adverse events. I always explain to my patients that a drug that does not have any adverse events is possibly not working, not doing anything. Because whatever you take, cannabis, a drug, a chewing gum, whatever you take, you put something in your body and there will be wanted and unwanted effects. The wanted effect is the therapeutic effect. The unwanted effect is bad. If you eat too much sugar, you will have decay in your teeth or diabetes. So whatever you put in your body, whether it's a drug or food, will have wanted and unwanted adverse effects. So there are adverse effects. And I find it almost reassuring, because the fact that there is an adverse event means the drug is doing something to your body. Otherwise, it's just a placebo. However, you have to be aware of these adverse events. And the older the drugs, the more follow-up we have, and the better we know the unwanted, the adverse events. When I started med school, we were not really aware of the adverse events. Now we know them, and we have learned how to control them. So you have to be aware of the adverse events so that you can give the patient an adapted prescription. For instance, if you give a big dose of any therapy to a patient, the patient will not feel well, and there will be many adverse events. So mostly when we introduce a new treatment, we give a small dose and we increase the dose progressively. I mean, it's our duty as doctors to check with you that the adverse events do not appear. We cannot simply say, oh, by the way, I wanted to tell you that I am spending a lot of money and my bank balance is close to zero. No, you have to take the time to talk to the doctor, and the doctor needs to talk to the patient, and the patient needs to ask the doctor about the adverse events. You don't sell your house overnight. You decide to do something, for instance, you go to the tobacconist and you buy a lottery ticket, or you eat more chocolate than you used to. I mean, you know, when you see something like that, you talk to your doctor, and maybe your doctor will decrease the dose or maybe switch to a different medication. In Parkinson's disease, we have several medications available to treat the symptoms. And you said it's not new, and that's what I find interesting. It is not new, as you just said. For a long time, doctors did not ask questions because they were not familiar enough with the adverse events. And there are hyperdopaminergic behaviors. And sometimes the patients themselves were ashamed and did not want to talk to the doctor and say, I'm spending my family's money, I gamble in the casino, or they had behavioral and impulsive disorders that they were ashamed of. And the research was so limited that we did not really know the adverse events. Now that we are familiar with the adverse events, now that the patients play an active role in their treatment and the doctors are much more informed, they ask the right questions immediately, we see that there are less adverse events. Now, if you start a protocol to look into more specifically Parkinson disease patients with behavioral disorders and impulsive disorders, you have to wait for a while before you have a sufficient number of people with real issues, because more people are being managed better, the therapy is more adjusted, and we see less serious adverse events. Obviously, for research purposes, sometimes it's not good enough because we have less subjects with bad behavioral issues, but at least it's a good sign because it means that our research is working. The longer it takes, the better in a way. The longer it takes to have enough people with serious adverse events, the better. It shows there was an evolution. I'll take a question online. We're going back. As you understand that we move from one topic to another, so we'll go back to the organoids. There is one question here. Why use organoids instead of human tissues? Not enough donors? No. I mean, obviously, it's not simple to obtain human tissue. And even if we have access to human tissue, it's extremely precious, costly. Could you tell us what kind of tissues you're referring to? Well, usually we have access to post-mortem tissues, but we need to make sure that we have enough tissue harvested exactly in the same area. We also have to have control tissues, but maybe control tissues coming from a patient who has a different disease, not Parkinson, but a different one. And that means the control tissues are not healthy. And, you know, the tissues are very precious. They're very valuable. And we use postmortem tissues to validate an assumption once we already have conducted the research. So organoids are helpful because they give us a possibility to work on human cells, but in a very controlled environment. In the experiments I showed you, we have organoids derived from control cells, and the cells where we prevented Parkinson's from expressing them are the same. The only difference is that Parkin does not express itself, so we can compare the two in a very strict way. That allows us to have more control, and it allows us to have even more tissues available. Thank you. We will now take a question from somebody in the back of the room. I would like to go back to something practical. A patient being followed by a community neurologist, not in the hospital, and who for the first time says, well, when the doctor talks about addiction and compulsive behavior, the doctor will make appointments every 6 months, but the patient will be ashamed, will be uncomfortable, and will not want to talk about it. And the patient will be afraid that the medication is being removed and he needs the medication and would rather not say anything about the compulsive behavior. So what about the pump? When exactly do we start thinking about the pump? When do we start saying, okay, if the medication is an issue, could we move to a pump? Our pumps may be not well known enough, and how is it going to work for people who have issues with their medication? Well, when adverse events appear, the first idea is not necessarily to switch to the pump, but the message I really want you to get across, and I want you to disseminate the idea around you, is that there are many different molecules available for symptomatic treatments. And there are 2 new pumps currently available. They came on the market in 2024. But for oral medication, if behavioral disorders appear, we decrease the agonists and we increase the L-DOPA. We never really stop the therapy overnight in the patient, and that could be bad. I mean, that there is a big threat with major stiffness if you stop the treatment overnight. So never, ever, never discontinue the treatment overnight. Talk to your doctor, and if you want to discontinue a treatment or decrease the treatment, do it with the doctor. That's why today we are mentioning the symptoms so that everybody remembers to talk to the doctor. There's no shame. I mean, symptoms are an adverse event, and you really have to talk with the doctor. The doctor could be your GP or your neurologist or someone else, the hospital doctor. Maybe you can talk to your GP and say, could you please contact my neurologist? I don't know what to do. And that's one thing. And the second thing is that you don't necessarily switch immediately to pumps. Switching to pumps is something that requires a joint agreement between the neurologist and the hospital neurologist, usually between the community neurologist and the expert center. We assess the possibility to switch to pumps, and maybe the patients will also talk to specialized nurses. We now have a new figure, that of specialized nurses, and they play an active role, and they are very helpful because they're close to the patients. Okay, I'll take one more question online for researchers we have here. It's a broader issue. Are research topics shared with other countries in Europe, in the United States, or in Canada? Is there any international collaboration? What's the role played by international collaboration in current research? Well, maybe 100%. In research, we never work alone. We work in collaboration. We have different approaches depending on the country. And we can share our results. We can share our results and research. Most research topics are collaborative. Even in France, the National Agency for Research funds collaborative research. We're never or rarely alone to carry out research. We would never really achieve anything. We would go around in circles on the same ideas. But we do have cultural specificities. And for instance, if the French were the first to work on deep brain simulation targeting subthalamic nuclei, generally speaking, the French clinical teams for other applications, other indications than Parkinson's disease, for instance, obsessive compulsive disorders. The French teams are defending the subthalamic target, whereas the Americans will give precedence to the anterior arm of the internal capsula. And sometimes they say it's the commons, but really that's the area they target. So there are cultural differences, but whatever, we are working together. It is collaborative research because that's what research is all about. We want to work to improve knowledge. So we help each other. And even sometimes, although we choose different targets and different strategies, we discuss them together. I mean, that's why we organize conferences and we share our results, we compare our results, and that will help us come up with a strategy which will be the most useful or the most efficient one for our patients after we have each tested different things in our respective countries. But sooner or later, we will converge and there will be publications in international publications. There will be papers published in international publications. There will be congresses. And we may sometimes decide to organize a joint research. And that's also a way for us to find the funding, because obviously it's very difficult today to obtain the funding if you're on your own. Well, on this subject, I really like to say that compared with when I was doing research myself, there is less competition. Okay, there is still some competition, but we're not fighting, working on our own and hiding our results because we want to keep the funding for ourselves and we don't want to share. I mean, it's still there, but I think there is now a trend with bigger sources of funding, international consortia, and it means that we have to join forces and bring together our knowledge. For instance, the ASAP project, the Google co-founder who has Parkinson's disease cases in his family, and he's giving billions of dollars worth of funding to research. So we're talking about collaborative research. It's not one researcher or one country. It's several countries, several researchers joining forces and sharing their expertise. And research has moved on.
Alors, il y a des programmes spécifiques où justement ils sont obligés de dire il faut collaborer, sinon vous n'aurez pas l'argent. Donc évidemment, là-dessus, on est tous copains et on va tous
We're all friends, you know. We're all going to postulate and form consortiums and work together. That's all well and good, but you have to be aware that we're in countries that finance research the least. And when there's no money, there's competition because there's not enough for everybody. And on the European level, we're ahead of programs that are very elite, such as the European Research Council, ERC. And these are programs that are going to be giving money to one person, 2 million for 4 or 5 years of research, and nothing for anybody else. So we're in a culture of, the scarcity of funds. It's not broken down equitably. That's the period we're in. And it's not that comfortable. So thankfully, there are programs, the ENR, there's a lot of programs that are collaborative, more than, just single teams. But the situation is complicated. I mean, this is not mutual perfect love in the mutual world, in a wonderful world. I think it's better. I'm not saying it's resolved. Thank goodness that France Parkinson has financed a lot. Let's take a question from the floor over here. Thank you for the presentations. Can you hear me? About mitochondrial aspects, you want to draw the parallel between stress and dopaminergic. If you have if you reduce significantly chronic stress, can you reduce some of the symptoms, or does it have an effect? If you diminish that.
Alors, c'est une super question.
That's a great question. This is part of the project of Christian that I didn't want to explain because we would have to add the whole notion of chronic stress. And so is it a one-off stress, a strong one-off stress, or a chronic stress? such as what I showed you. We're going to be stressing the cells just a little bit, but regularly for a long period. So we are currently doing that. What we're looking at are the answers to punctual stress different than a chronic stress? What are the differences? So that's the first thing. They are a bit different, which is what we're expecting. But in the case of Parkinson's disease, when you think that the cells are basically stressed already, so that would be a chronic stress. So the cells that had a chronic stress, do they respond not as well or better to a one-off stress? So basically, we're trying to see if cells that have been stressed for a long period, even just a bit for a long period, if they're able, when they are subject to a very strong stress, all of a sudden, do we need protective responses as in normal times? I can't answer you because that's what we're studying. Thank you. I guess we'll take a question on the front here. Hi. I have a question from a neophyte. I'm a daughter of a patient. I'd like to know, it goes back to the idea of funding. You summarized in 15 minutes years of research. We've understood that we'll not see the effects right away for patients. For each of you, what are the next steps of your research? And we say the next steps, not really technical. I mean, you're funding for what is to follow. And for France Parkinson, are we financing lots of research, or are we going to actually help research that's already started? I think the question is for me. No, it's for everybody, really. Just to say the choices of projects, we have a call for projects, either social or medical. We receive a lot of projects. I think we finance a lot of fundamental and preclinical research, preclinical research. We'd love to finance all of those subjects. We don't have an envelope that would allow us to do that. And we have a scientific committee of experts doing fundamental research, which represent just about all of the skills available in France, that will be choosing to select the projects on the basis of their scientific value, the level of innovation, and feasibility as well. So we classify these in order, and we try to hand out projects as the envelope is started. We had up to 2 million last year for research projects, which is a lot. But it's true that for the moment, we have projects, but we don't necessarily have an automatic continuity of financing. We have that in mind. If a project has interesting results and promising results, do we then grant additional money for a year more so that they can move forward and get more results so it can be more transitional? So we've had this in mind for a while, and we have to implement that. But we're obviously thinking about this a great deal. If I could add something, personally, we're all working on fundamental research. Maybe a bit more clinical for my colleague, or we're pretty far away from medication that's going to be used to treat people. And public research doesn't have the resources it costs a lot of money to bring medication to the market. In the lab, I had a student on a thesis who worked with me, who wrote a few articles, and who was able to get a therapeutic in case of chronic pain outside of Parkinson's, and we derive value. We got it patented, and a researcher put together a startup whose objective was to get private funds, private investors, in order to develop the medication. Unfortunately, that's the way it is. You can't develop, in a public context, a new medication. We need private funds. Unfortunately, we live in a capitalistic world. So there are systems and processes that are like that require many more resources than I wanted to respond about the continuation of my project, which for some it might seem pretty far off when you say we're working on something like and the social context. We still see an involvement, a potential involvement, and possible side effects to detect. And since sociability was never studied in Parkinson's patients, how are they affected by deep cerebral stimulation? There's been no study on that. So it's not necessarily me that's going to be setting up the study with clinicians. But the fact that we are showing that in animals, and that's what's great in our research community, clinicians also look at what we're doing on animals. At least some of them do, and they're going to be curious to question their patients and look at this, and we can see in a few years, hopefully, development of research that could be more interested in sociability in Parkinson's, and how is that affected or not by stimulation. I'd like to get back to the aspect cited of valuation, deriving value for startups. Sometimes we received emails saying it's scandalous. Startups are created because they want to make money. You have to realize that it's a change in the area of research. The industrials were doing research and preclinical right to development. They're financing the whole thing, and it costs a lot of money. With a lot of medication that are lost, that fall by the wayside. And these industrials, in the last decades, they've just withdrawn from fundamental research. So they depend on academic or financing. So there's this kind of no man's land between the preclinical results and the industrial who has to be able to put that on the market. And that can only be handled by startups. The existence of biotech startups is indispensable to get from that fundamental research over to actual application in the clinic. And unfortunately, all the startups that have brilliant ideas don't always get the money. You have to interest the right people. You have to get the financiers. And I think this is a phase that today is indispensable to research. It really makes that transition feasible from fundamental preclinical to more applied and clinical that would be taken over by industrials and who can invest millions and billions to bring it to the market. So I think you have to bear those issues in mind. I think we've reached the end of our time. We can continue 5 or 10 minutes if there's still questions. I think there's one question here. You've been sitting down for a couple of hours. But yes, sir. I'd like to know how much is affected to research. And the park is to cure the disease. What share of the amount? And I'd like to know where AI stands in research on Parkinson's. And the researcher in Tours, in France, who was proposing a medication for diabetics that stopped degeneration.
Alors, donc, il y avait deux questions.
There were two questions. And I think we'll let you go. About the two questions. The first question was, if you're talking about preclinical or clinical, in actual trials, every year there's an article that's published on the pipeline of studies that are being done with a type of approach, symptomatic or whatever, neuroprotection and various mechanisms that are targeted by a researcher who does the review every year of that pipelines. Globally, it's like 50-50, maybe 60-40. There's an effort in both areas, in both types of approach. In symptoms, there's also people that are interested in motor symptoms or non-motor symptoms. Now, for the anti-diabetic medication, you're talking about lexinitide? Lexinitide? There are two anyway. Lexinitide. So this is the study that we participated in.
Moi-même, je veux qu'on arrête.
I think we have to stop. There's a study in Great Britain with a molecule that was positive in phase 2 and negative in the following phase. And in France, there's a study in the previous that was positive, and the new phase isn't done yet. So the mystery is all still very much there. You can't actually, if you're not diabetic with Parkinson's, you can't be prescribed that medication because the efficacy and the safety profile has not yet been confirmed for use with diabetics. So you'd have to take it in the framework of a clinical research that protects you and protects them. Well, you might have to choose a specific molecule. And you have that in France and in Great Britain, but it's not yet confirmed. Thank you so much. We're going to have to stop at this point. Thanks for everything. I'd like to thank a few people, so don't leave right this second. First of all, I'd like to really thank our speakers for coming today. They're not all from Paris. Some are from Marseille, Bordeaux. So thanks for having to simplify research for people and understanding. I'd like to thank the Foundation for Research Cerveau that we've been working with for many years and who also shares with us the choice of projects. And also, you have right, let's applaud this. And Dim Sibrin, who supports us for this day. And I'd like to thank all of the France Parkinson team who are present today. All of the staff are here and our committee. Without them, this day would not have been possible. So thanks for everything that they did. I'd like to thank our committee because France Parkinson, our staff in Paris, but these are there are 900 volunteers in France that help us to do actions locally throughout the whole territory. This is really the live force of France Parkinson. So thanks to our committees in France, and some are listening to us, and I think they really are worthy of your applause. Our events would not be possible without the team that helps us, called Pointe Noire, who've been helping us for 4 years now. And as I say, each time working with them is working without stress, because even when there's problems, there's no problem. They always have a solution. You don't see them, but they're there, and I'm so thankful for them. It's always a pleasure to work with them. It's important. And I would like to thank all of our members and donors. Without them, France Parkinson would not be there, and we would not finance research. So if I had to present research, it's thanks to our donors. We hope that we'll all be unemployed soon, that we'll have found final solutions for Parkinson's, but awaiting that moment, we'd like to thank donors who will let us help research move forward. So thanks to our donors. Before we close, we're going to have some water and some biscuits. I think it's going to be a good time to stand up and get some physical activity. So we'll give you a glass. It's the 40 years of last year. It's a gift. You can keep the glass, take it home with you. Think of us next time. You might be some cookies. And there's a discovery area. If you haven't seen it before, go there now. The young researchers are there to respond to the questions you have. Some of them have subjects that we spoke about today. So take advantage of the space is open. Come and have a look and see you next year. Thank you so much.